So now we have talked a little bit about how to analyze our data set in many ways, from the beginning to the end. And another thing that you should always consider, and this is also a new lecture that we haven't had before in the course, and we have a little bit of the exercises as well, is some online repositories that you should be aware of. There are many, many, many. Some of them we already mentioned, gene ontology, carrier pathways, and so on. But those are related to annotation of genes in that matter. But now what I want to mention is like how you get data from others that others have published, or how can you deposit your data, or where should you put it and why. So this is also very brief, but it's just good so you have an idea what is ahead of you. Maybe now that you have RNA-seq data analysis, you will probably put your data in one of those repositories afterwards. One that you have seen all the time during our lectures before, in Roy's presentation specifically, and the ones from Johan, is Ensemble. So Ensemble, you can go there, as this is the main page, and check which species you want. And maybe you can type a gene or not, or you can simply click on mouse or human. And there you have all the curated genomes that are already sequenced and annotated. And it's exactly from that web page. If you just, for example, look in mouse, download DNA sequence, this is the sequence that you used in your exercises that we selected as to download from UpMax, but actually that sequence came from this website. The same thing from the GTF. So Roy also showed in another way how to access that. And this is just via the web browser. You can also access in this way. And there are many other formats, so the GTF or the GFF3 that we already mentioned as well. So yeah, so briefly, it's just to show you, oh, I have a different mouse genome. What do I do? Or I have another species or Salmonella. You go here and download that species. Another one, and this is one of the most known repositories, is the GEO, so Gene Expression Omnibus. And that one comes from the NIH in the US. So it's a repository that is in the US. So if you have, for example, a paper in this one, so we can reuse public data that is deposited there. So for example, in here, we're using many data sets, and they are named with the GSE tag and then a number. So this is the identifier for that data set. And each data set, when it's deposited, is usually associated with a paper. And also, for example, in the same paper, there is data availability, and then all the raw data generated here were deposited in the GEO under a session number. So this one. And then you can also just go to the session number on GEO here. And as the link shows, also the GSE ID. So if you just go to the web browser on GEO, this is the web page that you'll see, and then you can type keywords here, either the name of the paper even, or the person that deposited, or in this case, the GSE code from the paper that you're interested in. Of course, if they deposited it here, which is very likely, but not always the case, but it's very likely. And then here is basically where we type the GSE, and then you can see this is the GSE with this number. And here you can see when it was released publicly, and then the name. It does not need to be the name of the paper. It's just the name of the data set. What has been done in this data set, which is exactly the same as we have seen in the paper, or like day 0 versus day 7. This is exactly this data set. So the experimental design, so the mice were treated with DSS, induced colonic inflammation. 2 to 3 animals were sacrificed at different time points when the colonic tissue was collected. So we know which tissue it comes from, for example, and other more details about the experiment. So we know who deposited. We know there's a publication linked to this one. So always when you use this data set, then you mention it, you cite it in your paper, just like as I mentioned in here. So you have the different data set. Here I'm just showing relevant information that I used from them. And then here I'm referencing those data sets. So not only publishing your data is good for your quality metrics because your paper will get cited because you have a data set, not only because you have done an amazing job in analysis to uncovering a new pathway, but also because you allow your data to be reused for others, for many other purposes. And that's actually really good. So for example, here now we have more information in the page if you scroll down. So every sample will be assigned to a GSM tag and then some values. And then here's like the title of the sample, DSS 0, 1, 2, and 3, which is exactly the samples names that you probably have in your dataset that you have been using. The technology used is usually indicated here. So in the GO, you'll have microarray, RNA-seq, and other sequencing technology. So ATAC-seq also probably will be here, and many others. We also have what is called a SRA accession. So I'll talk about SRA in a minute. But this is the link to the raw data. So all the FASTQ files from this experiment is also deposited. Since it's a mouse, we don't need any kind of protection in terms of like patient sensitive information. So all of it is freely available, and is also where we got the FASTQ files from this. Even though it's my paper, I don't have the FASTQ files anymore. Everything is deposited there. So I just download them again. And the metadata of this matrix is also here, is what called series matrix file. And it comes in different flavors, either soft, min, formatted, or this series matrix. And you will be able to download later in the exercises. And this case is a text file that you can actually open with Excel. And then there are sometimes some processed count files that are shown in here. So the raw here is the combined files from each sample that I'll show you in a minute. And these processed ones could be, for example, here it's the Callisto counts that I used. Callisto you'll see a bit later tomorrow, but it's exactly the same, the raw counts that I have in the data set. And here I'm showing the log2 counts per million. So I also deposited the normalized data that I used. After all the transformations, I can also get this one and run the analysis from that if I don't want to normalize it again. And then if you press, for example, click in one of those samples here, you'll have the sample number here. And then you can see that this sample is associated with this gene, this series. And for each sample, not here, you will not see the raw FASTA file, but you'll see, for example, the counts for that file, the individual counts. And if I'm not, yeah, so those counts were also used for creating this course, for example. Any questions about this? Or
do you know how it usually works with human samples? Like, are the FASTQ files ever uploaded, or is that usually just counts?
Yes, so usually we'll upload the counts to GEO, and the FASTQ files you need to upload it to a restricted server, and that is called ENA, the European National Archive. In this, I'm talking about Sweden. So the data should never leave Sweden, and Sweden has a node within the European National Archive to store human protected data, protected data containing human important information. And actually, there's one slide answering that question as well. So SRA is the sequence read archive, also from the NCBI or the NIH, where all the raw data is deposited. So if I submit my data to GEO, automatically they will deposit the FASTQ files that I submit to them directly on SRA because they are connected. And that makes things a lot easier if you work with mouse. So then what I can do is type the SRA tag that I saw in the GEO, or just click on the GEO, and that will show me this page. And this page, you can see the same GSM, which is the sample IDs that I have on the GEO. And then you have the sample names here. So they take care of all those small details for me. And then if you click on this sample, for example, then you can see some metadata, and they also run other QCs as well. So here, for example, they can see that the length of the FASTQ files, how many reads there are in here, the size in base pairs or the size in megabytes, the percentage content that we saw in the metric QC when it was submitted. 100% of those reads are single-ended 50 base pair reads. And then you can see many other things. So it's a Lumina platform and so on. And then you can also click on the data access tab. And there is the access via the NCBI. And this is just a link. If you click it, it will download the FASTQ file directly on your computer. Or you can write a bash code, wget, wget, or curl o, and then this name, that will download the file for you. If you know, for example, like the sample ID, the GSM value, then you can basically download all the samples very automatically. And there are other ways also to connecting to get that data. The European version of SRA is called ENA, so European Nucleotide Archive. And that is the website, so very similar to SRA, but in Europe. And the good thing is that the experiments that are open on the US are also available in here, even though we say, it's in the US, but we still have access to it. It's not that, it's Europe versus US. No, it's just we still have access to this. And in this case, I can type my GEO number, which is from the other database in here. And that will show exactly the same experiments that I have there and have the link to that page. So it doesn't matter basically where you are looking for. Those databases are the two major ones, or that I mostly aware of and that I use. And they are linked together. So yeah, use them. But how you deposit your data then? I mean, I will not show exactly step by step how to do that. If you go to GEO and then submit, and then there are the instructions in there. But things that you should consider, and just to explain. So what should we deposit? So you should deposit all sequencing data, raw, even including the FASTQ files, all the metadata or any processed information counts that you might have, as long as that doesn't interfere with any like identifying patient information or like SNP for genomic data, that should not also deposit. But in case of counts, estimating the genes or the transcripts, so that data can be deposited. Why? You should deposit to allow others or yourself also in the future to reproduce your results or use it for a different purpose. For example, using this course. And when you should submit the GEO before submitting your manuscript. Most journals are now requiring that. So when you submit, you can have a protection for your data. So the data will not be public. Only you can access it via a code. And that code is kind of a password. And then you can send both the GEO ID and the code to the reviewers. So the reviewers can see that you have uploaded the data. And they can actually run, if you have some analysis steps, they can rerun the analysis steps on their computer. Oh, actually it works, and so on. And then once the manuscript is finally accepted, then you can have an option in GEO release to the public. And that's usually what you should do. So when we did the submission, the reviewers actually asked us before that. So we add this information on the manuscript. The data will be deposited under this gene ID. And once the manuscript is accepted, the data will be released. So if you have non-human data, so anything other than humans, it's very easy. So then you can either submit everything to GEO or ENA or any other database, including the raw FASTQ files, metadata, and other processed matrices. But if you have human samples, then you should first, for example, in the case of GEO, which I'm quite used to use now, you email GEO, specifying that you have human samples that you want to send, and then you submit the raw FASTQ files and metadata to ENA, which is the European, the Swedish version, as well as with restrict access. So this is the raw information of the human, and all like patient IDs probably will not submit. And then to GEO, you need to process, have the processed counts and the metadata. And then you send also the link to your deposit data on ENA. And that link will only be accessible if the two universities where the data is hosted will agree to that. So the data is protected. That's what I mean. Once you deposit them on ENA. So only people that you give access to will always will have access to the data. If you want to have that restriction, I mean, for human samples, you should have that restriction.